Well, thank you very much for Karen for the invitation and all the organizer. My name is Alassane Mbengue. I'm based at the Institut Pasteur Senegal, where I was born and raised. And then after my after my PhD in France, I came back to Senegal, where I'm leading the antimalarial resistant team since a few years back. And then my journey into 3 continents, I said that I was born and raised in Senegal, moved to France for my PhD. I did a postdoctoral studies at Notre Dame University, and all I used it microscopy and during all my studies to better understand or decipher the mechanisms of, I would say, the survival mechanism of malaria parasite. I am currently in Cape Town, where we're having the MIM, which is the Multilateral Initiative on Malaria. And then what is the trends in malaria was a lot discussed with the climate change, meaning that, you know, if you don't move quickly against this disease, there will be more increasing cases of malaria around the world. And this is a map that I was trying to understand. You know, this have been drawn over the last years, showing all the problems or identified challenge, not only in Senegal, because we're talking about impact of human movements on parasite transmission, and then this is not clear to us how this is to impact, you know, malaria incidence or not. So we better quickly develop new tools to understand the pathology of malaria. So as you would see, you know, during all my studies, my work focuses on the asexual live stages that last for falciparum 48 hours. And then here to track artemisinin resistance is the main problem, not only in Africa, but often emerging from Southeastern Asia. We have 3 ways to track artemisinin resistance. And you would see that all these techniques are linked to microscopy somehow. The first thing is that when you do the experiment in vivo, you have this technique called like the parasite clearance, and then in which you determine the parasite level upon transmission, meaning that in the field, either you determine the parasite or the type, species of falciparum that is infected patients, and then we will talk about that in the field. There is a real need to develop microscopic tools in order to identify those 5 human species. And then ex vivo or in vitro, there is this RSA, you know, commonly called the ring survival assay, in which you have parasite, you synchronize them, because artemisinin does affect early ring parasite, and then you shoot them with a pulse of 700 nanomolar DHA, and then you wash out, and 72 hours later, you count the parasitemia. And again, here you would need like microscopy scales to estimate the RSA, which is like the resistance level. And then in addition to that, we're tracking calcium, which is the validated antimalarial resistance that you're seeing here. And then with Amy Christine Bay, who's at Yale, you know, we have this protocol or this method, working method, in which we combine both the field clinical work that I mentioned earlier, in contact with the population that are infected with malaria, and in the lab we're trying to develop skills, you know, sequencing methods, and also CRISPR-Cas9 that I will talk about in order to understand the functional relevance of mutations that have been seen. So I said it before, you know, thanks Sabrina, you mentioned many things that I wanted to start with to talk about malaria, but here if you focus on the asexual blood stages, the parasite need to export hundreds of proteins. And in these schematics that I made a few years ago, the parasite protein from the ER has to cross several membranes in order to reach the morus cleft, that is red membrane that are close to the infected cell membrane, and in which are attached other proteins that allows the parasite PFMP1 encoded by genes, and then this allows the parasite to bind to microvessels, and this is in human the causes of severe malaria, causing like a lot of death. And then all these modifications that you're seeing here on your right side are due to malaria exported protein. So the exportum I will show how microscopy have been used during my thesis to kind of understand the exportum that is tightly linked in conclusion to the virulence mechanism of malaria. So something to keep in mind is like, the parasite, the infected erythrocyte have deformability properties also. So this is physics. And then if you combine, in order to understand the parasite dynamics within the erythrocyte, there's also something that is relevant, which is like the cell deformability, the rigidity. And this is a paper that was done during my PhD in France, where my farmer boss worked with some physicists, and then they show that, malaria releasing the merozyte is not only a burst, but this is a synchronized parasite in which you have complicated mechanisms such as the curling and the blocking that allows the parasite to be released. Not all the merozyte are released in the blood. And that makes sense because in the clinics, you never have you can't reach such high parasitemia because the parasite understands that if he kills its host, it will die. So this is how the microscopy could impact understanding the egress in malaria. But something also to keep in mind is we need to develop some biochemistry tools in order to understand the proteins that we often targeting to understand the exportum, and also generating transgenic lines. And I think that Sabrina showed a GFP Kelch13 protein that could be used to identify the mechanism, not only the mechanism, but the exportum as well that are involved. Those proteins are also involved. So membrane attachment or membrane properties are also important in addition to the skills that we talk about, the microscopy skills, I would say. Now, if you go to my other side, this is just summarizing the different stages of the parasite during the infected erythrocyte. I won't talk about the invasion. It was nicely done, but here again, what is important is the parasite within the erythrocyte pass through different stages, a ring, trophocyte, and a schizon that Sabrina has shown earlier. But what is critical is during the 48-hour life cycle, the parasite is permanently growing inside, digesting the hemoglobin, and while doing it, the parasite is exporting hundreds of proteins at the edge of the infected erythrocyte, and this would cause the cytoadhesion of BFAMP1. And so this is also critical to understand. I said it before, generating transgenic lines or recombinant protein that are either tagged to GFC could help. So, here I'm just trying to show that, if you don't have proper antibodies, you could generate GFP lines used to address the exportum issue using confocal microscopy. And this is an example where I did a GFPE61 protein, which is a Morris protein. And this is how it shows up within the erythrocyte. These are two parasites that have invaded the erythrocyte. And you can see that in these ghosts, what we call ghosts is when you remove it from the infected erythrocyte, you can identify the Morris protein. And this could be served when you have good markers such as PFAM, SBP1, that is a marker of the Morris you can clearly identify where your protein or targets are being relocated during the erythrocyte. So because of the matter of time, I can't talk about all these biochemistry protocols that we developed in order to fractionate the infected erythrocyte. But during my postdoctoral work also use similar methods to identify what are the targets for artemisinin resistance. And I'm using a pull down, which is a consequence of the localization of these proteins within the erythrocyte. We were able to fractionate, but also to pull down proteins. And this is how I identified the PF trick that was later shown to be involved in artemisinin resistance for the first time. I identified during my PhD. So similar method was used in the artemisinin context, and that allows me, so here you can see how I'm using microscope in order to purify or to understand the mechanism of artemisinin. And this paper was published in 2018 when I left the US. So this is also a summary how I did combine, because I had these relocation or these mechanisms that are involved in artemisinin resistance. Here you can see when we compare ANL1, which is like sensitive to artemisinin resistant, versus a parasite that came from Cambodia that is resistant. We saw the amplification, and in blue you have the W, of course, and then these are around the same size parasite, and you can see that there was a huge enrichment of these PI3P, and that was used to better understand the artemisinin resistance method. Next, I use electron microscopy data. We blinded our sample, ship it to another lab, and we identified clearly that in the artemisinin resistant mechanism linked to calcium, there were an amplification of PI3P, and that was also published in 2015. So, sorry again for Zeiss, but one of the microscope I enjoyed the most was Delta Vision, and I was quite happy to find it in South Africa last year when I went with the CRIP funding. And just to say to the community that there are microscopes available in the continent, and they either in South Africa or in Ghana, but please feel free to be in touch with either Karen or Mike in order to get access to those microscope facilities. So what is relevant now is we now know that there are clusters of parasites. This is a map of the work done by our colleague at MRC Gambia that are showing that we have cluster of parasites in each country. What does it mean? It means like a lot of the labs right now are using either 3D7 or NF54, and these parasites, they were isolated in 1979. And we now know that because of the human pressure, because of the heterogeneity of malaria transmission, there is a real need to consider developing tools to understand that are parasites that are currently circulating in each of these countries that are facing malaria challenges. So in other words, the reason why I have been pushing and then working with these guys to develop microscopy in Africa is like there is a real need to develop these skills and in order to better understand. So the question is like, are we facing the same mechanism for artemisinin resistance when we're comparing it to Southeastern Asia? I think that there would be like a shift in these mechanisms. So this is Lati, who's a postdoc in our lab working with Dr. M. Christine Pei. He's doing a good job. And in the field, we have also this need to develop skills to identify these Plasmodium species, because in addition to falciparum, we now know that Malaria and Ovale are also circulating. And then if you go to the field, there is a training from, at least in Senegal, from the National Malaria Control Program. These guys are the ones who are training these clinical healthcare workers, but often they don't have microscope or they are not fully trained to identify or to count the parasite, identify them, I would say. And that's also a major problem in Senegal. This is an example of how to integrate CRISPR-Cas9, and this work was done by my PhD student who went to Senegal, who went to Montpellier, and she did like an amazing job, because this is the first time we've identified one parasite with a Southeastern Asian mutation, the R515K, and she was able quickly to develop this tool in Montpellier. And then again, I think that we all understand that, when you're tracking artemisinin resistance, or at least if you want to develop fundamental science, you need to have this lab where you keep growing parasite, doing your biochemistry and also molecular testing. And again, here you would need microscope in order to estimate, to grow your parasite, and also make some tests again and to understand antimalarial resistance. And this mutation again was found in Eritrea recently. But before this paper was out, we were the first one to generate these transgenic lines. And also we found out that in red here, you can see that there was an increased RSA, and this mutation was found in central regions in Senegal. And in addition to that, we use another mutant of kel-13 that was shown to be associated with very low frequency of artemisinin resistance still in Eritrea. And you can see that in yellow that there were like very low RSA level, which is like the measurement, the in vitro measurement of artemisinin resistance. And that was confirmed recently by another work done by BJ Mena and also David Fidok's lab, showing that this mutation, finally, the R622Y mutation, does not confer. And you can see that not only it does confer a low ring survival assay, but if you compare DD2 and NF54, two parasites that have different background, you can see that in NF54, we have like a higher RSA level. What does it mean? It means like, again, we need to develop like quick tools in order to understand those clusters of parasites that I've shown earlier in different countries that have like different adaptation methods. And this is to me why we would hopefully move to immunofluorescence again to understand the long non-coding RNA for PFA and P1 that I've shown earlier that are involved in the adhesion. So thank you very much. I just want to highlight the need again and talk about the need again of microscopy in the continent. After ABIC, I was working with Yao Anibe, a colleague and friend of mine in Ghana. We now have this consortium where we're organizing microscopy tutorials or classes with students, you know. So I would highly ask West African, you know, students or to apply for these fundings, either ABIC or Wambian. And then these are my other activities in this high school. We've been there using microscope, showing it to high schools. And this is a documentary. I'm often making like some documentaries. And as an artist in Senegal, I'm trying also to have the artist community to join forces with to combat infectious diseases. And this could have been done without this international collaboration where I have, you know, friends now all around the world, I would say, and then students who are doing an amazing job in the field and also in the lab. We're pushing hard. And then just to say that it's not that easy, but I believe that with all these collaboration, you know, we could, put like, implement like a cutting edge science, and that would be critical to combat these infectious diseases. So thank you very much. I am done. Appreciate it.
Thank you so much, Alassane. I really appreciate it. What an interesting talk.